Bismillahirrahmanirrahim. Embryo culture. What is embryo? A seed plant embryo is part of a seed consisting of precursor tissues for the leaves, stems, and root, as well as one or more cotyledons. The young sporophyte of a seed plant, usually comprising a rudimentary plant with plumule, radicle, and cotyledons. What is embryo culture? The embryo of different developmental stages let us make it more bigger. The embryo of different developmental stages formed within the female gametophyte through sexual process can be isolated aseptically from the bulk of maternal tissues of ovule, seed, or capsule and cultured in vitro under aseptic and controlled physical conditions in glass vials containing nutrient, solid or liquid medium to grow directly into plantlet. Culturing method. The general method of embryo culture follows the following steps. Pluck healthy and mature fruits from the field and wash thoroughly in running water for about an hour. Surface sterilize with 0.01% between 20–15 minutes. Rinse seeds several times with distilled water and finally treat with 0.01% HgCl2 solution for 10–15 minutes. Finally rinse it for 6 times with sterile distilled water. Sterile distilled water. Break seeds aseptically and isolate the embryo. Culture embryo on callus proliferation medium. Supplement the basal medium of Morashi and Skoog 1962 with different combinations and concentrations of sugar, vitamins, hormones and other growth adjuvants for callus proliferation and shoot regeneration. Incubate the cultures at 22–25 degrees centigrade under a 16-hour photoperiod of 2000 lux luminous intensity. After 2 weeks of inoculation, the embryo begins to swell on callus proliferation medium. Distinct callus growth is observed after 4 weeks. After 8 weeks of inoculation, transfer the callus on shoot regeneration medium. Within 4 weeks of transfer into second medium, the callus turns green and produces soft, spongy tissue. Some of these tissues are differentiated into embryoids. The embryoids produce cluster of budlets when subcultured onto shoot regeneration medium. The budlets grow into shoots and produce 2 to 3 leaf appendages within 12 weeks. Thereafter, they are separated into individual shoots and then subcultured into a fresh medium of the same composition until shoots develop. Types of embryo culture. Mature embryo culture, embryo rescue. Mature embryo culture. Mature embryos are isolated from ripe seeds and cultured in vitro. Mature embryo cultures are carried out in the following conditions. When the embryos remain dormant for long periods, low survival of the embryos in vivo to avoid inhibition in the seed for germination, for converting sterile seeds to viable seedlings. Embryo rescue. Embryo rescue involves the culture of immature embryos to rescue them from unripe or hybrid seeds which fail to germinate. This approach is very useful to avoid embryo abortion and produce a a viable plant. Wild hybridization involving crossing of two different species of plants from the same genus or different genera often results in failure. This is mainly because the normal development of zygote and seed is hindered due to genetic barriers. Applications of embryo culture: prevention of embryo prevention of embryo abortion, overcoming seed dormancy, shortening of breeding cycle, production of haploids, overcoming seed sterility, and clonal propagation. And now we'll move towards our next lecture. Meristem or shoot tip culture. Meristem. Meristem is the tissue in most plants containing undifferentiated cells, meristematic cells found in zones of the plant where growth can take place. What is meristem culture? Meristem culture is the in vitro culture of a generally shiny special dome-like structure measuring less than 0.1 millimeters in length and only one or two pairs of the youngest leaf primordia, most often excised from the shoot apex. Principle: the excised shoot tips and meristem can be cultured aseptically on agar solidified simple nutrient medium or on paper bridges. Dipping into liquid medium and under the appropriate condition will grow out directly into a small leafy shoot or multiple shoots. Alternatively, the meristem may form a small callus at its cut case on which a large number of shoot primordia will develop. These shoot primordia grow out into multiple shoots. Once the shoots have been grown directly from the excised shoot tip or meristem, they can be propagated further by nodal cuttings. This process involves separating the shoot into small segments, each containing one node. The axillary bud on each segment will grow out in culture to form yet another shoot. Remove the young twigs from a healthy plant. Cut the tip 1 cm portion of the twig. Surface sterilize the shoot apices by incubation in a sodium hypochlorite solution, 1% available chlorine, for 10 minutes. The explants are thoroughly rinsed 4 times in sterile distilled water. Transfer each explant to a sterilized petri dish. Remove the outer leaves from each shoot apices with a pair of jeweler's forceps. This lessens the possibility of cutting into the softer underlying tissues. After the removal of all outer leaves, the apex is exposed. Cut off the ultimate apex with the help of scalpel and transfer only those less than 1 millimeters in length to the surface of the agar medium or to the surface of filter paper bridge. Flame the neck of the culture tube before and after the transfer of the excised tips. Binocular dissecting microscope can be used for cutting the true meristem or shoot tip perfectly. Incubate the culture under 16 hours, light at 25 degrees centigrade. As soon as a growing single leafy shoot or multiple shoots obtained from single shoot tip or meristem develop root, transfer them to the hormone-free medium. The plantlets formed by this way are later transferred to pots containing compost composed and kept under greenhouse conditions. Here is a potted plant, and then we have magnified view of shoot tip, excised meristem placed on culture medium, and new shoots, a callus tissue, new shoots and callus tissue, rooting and conditioning of transplanted shoots, and hardened plants. Importance of shoot tip or meristem culture. The uses of shoot tip and meristem in tissue culture are very varied and include mainly virus eradication, micropropagation, and storage of genetic resources. Virus eradication. This technique is also valuable for the maintenance of carefully defined stocks of specific varieties and cultivars in disease-free state. The size of the meristem explant is critical for virus eradication. Micropropagation. Asexual or vegetative propagation of whole plants using tissue culture techniques is referred to as micropropagation. Shoot tip or meristem culture of many plants species can successfully be used for micropropagation. Storage of genetic resources. Many plants produce seeds that are highly heterozygous in nature or that is recalcitrant. Such seeds are not accepted for storing genetic resources, so the meristem from such plants can be stored in vitro. Thank you.